Bismillahirrahmanirrahim. Assalamu alaikum students. Today in this lecture I am going to discuss plant propagation, their definition, types and methods. And this lecture contains details about asexual type of plant propagation. So let's start our today lecture. Plant propagation is actually the process, or you can say it is a technique by which new plants can be produced by existing parent plant. Plant propagation is actually technique of making two or more plants where previously there was one. So by using various methods we can produce exact replicas that perpetuate much loved varieties, or we can create entire new plants that may become favorites of the future. Generally, we can define the term propagation as controlled reproduction of plant by a man in order to perpetuate a selected individuals or group of individuals which is having specific values to him. So we can use this technique for the multiplication of any plant or any individual which we need, or which we like to multiply. There are basically two types of plant propagation: sexual propagation, which is recombination of genetic material to form uniquely genetic individuals through the formation of seeds, and the second is asexual reproduction, or the asexual or vegetative propagation, use of vegetative organs to create plantlets genetically identical to the parent plant or clone by several different procedures, like by cottage, by stem cutting, by grafting, by budding, by layering, by division and by separation. So let's talk about each of them in detail. Asexual reproduction, or the asexual or the vegetative propagation, both the term is synonymous to each other. Actually, vegetative propagation refers to the multiplication or perpetuation of any plant from any vegetative parts. Any vegetative parts means the multiplication of plant happens by any parts of any vegetative parts other than the flower, other than the reproductive part of the plant. Flower is considered as reproductive part of the plant. That's how seed is the resultant of reproduction, and by the seeds sexual production occurs, and vegetative propagation occurred by the any part of the plant other than seeds. So by stem, by leaf, by root, by modified stem, by leaves, by modified leaves, we can multiply plants. Different forms of plant, different species of plants can be propagated by different parts of the plant. So this method actually involves the production of new plants using only the vegetative parts of the parent plants. So they are called plant propagation. This form of reproduction produces plants that are identical, that are very identical to their parents. There is no variation develops by this method. And asexual propagation may be faster than the propagation by seeds. Germination and growth of seedling is often slow compared to the propagation by cutting. So this process is considered fast method than sexual reproduction or sexual propagation. advantages of vegetative propagation. Students, before starting different methods, procedure and techniques of vegetative propagations, we will discuss its main advantages. The two main advantages we have discussed, like they produces or propagate plants as their parents. So they produce clone to their parents, identical to the parents. Their progeny are true to type like mother plants. And the second big advantage is they produce or propagate plants or multiply plants by this method is very fast. This method is very fast way of propagate plants or multiplied plants by this method than the seed or than the sexual propagation. So we will discuss here some other advantages, like vegetative propagation is the only alternate way where no seed is able to grow or no seed is available, no seed is formed, or germination of seed is very slow, or no viable seed is also formed. For example, banana, right? Next example is pineapple. Another example is roses. And one of the example is seedless grape, where seed is not produced but the plant can be multiplied easily. So certain rootstocks has the capacity of resisting or tolerating the adverse environmental factors such as frost and adverse soil factors like salinity or alkalinity. For example, frost resistance in frost resistant Fonseris trifoliate or trifoliate orange can be propagated by this method. Or the second example is Rangpur lime. So the ability of certain rootstock of resistant pests and diseases can be advantageously expected by this method. So this method can be effective. The next advantage is by this method generally drafted plants in nature is produced than the seedlings. Draft trees facilitate pruning of spraying or harvesting easy, and more number of plants can be accommodated in a unit area. To replant an undesirable existing tree, either with reference to its quality or susceptibility to pests and diseases, they must be better. The defects can be overcome easily by vegetative propagation through the grafting or budding of desirable section of the resistant tree by top working techniques. So many plants are propagated by vegetative means because the speedy, easy of multiplication. Novelty can be developed grafting for or budding on single plants, many varieties, for example roses of different varieties, to convert inferior varieties in superior varieties. Side grafting in mango is possible by this method. So you can see here apple dwarf plant in pot and rose variety. As any method, vegetative propagation also have several disadvantages as well. Like the plant is not vigorous and long-lived. They are usually short-lived plants. No new varieties are evolved or developed by this method because variation is not developed. Variation is only developed, or variation development is only possible by the sexual reproduction or by the sexual propagation, or the propagation by seeds. These methods are expensive and laborious, and they require most of expertise, most of labor. So this is expensive and laborious method, and they take time and expertise as well. Generally, the vegetative propagation is actually the growth and development of a plant by asexual means. Asexual means the reproduction in which only one parent plant is involved in the process of propagation. No sexual partners is involved. So variation is not developed. The development occurs through the fragmentation, means the segmented parts like stem cutting, like leaf parts, right, and regeneration of specialized vegetative parts like budding, like tubers, is developed by this method. So the development of plant occurs by the fragmentation and regeneration of a specialized vegetative parent plant is the vegetative propagation. Many plants that produce asexually are also capable of sexual propagation as well, but to maintain or to save time, asexual propagation or vegetative propagation is also used. Here you can see tubers of potato by vegetative means. Vegetative reproduction actually involves non-sexual plant structure, whereas sexual propagation is accomplished through gamete production or subsequently fertilization. And in vesicle plants such as mosses and liverworts, vegetative reproductive structures including gamma and spores, like are in vesicle plants, like in pteridophytes, especially in gymnosperms and angiosperms. Vegetative reproductive structures include root, stem and leaves. Vegetative propagation is made possible by meristematic tissues and commonly found within stem and leaves as well as the tips of roots that contain undifferentiated cells. And these cells actively divide by mitosis to allow widespread and rapid primary plant growth. A specialized permanent plant tissue system also originate from meristematic tissue. So meristematic tissue is developed and that produce new cells. It is ability. The vegetative propagation is the ability of meristem tissues to continuously divide that allows for plant regeneration required by vegetative propagation. So vegetative propagation is one of the best method in gardening or ornamental plants. So now let's talk about the types of vegetative propagation. There are lot of methods of vegetative propagation, but it can be divided into two main categories: natural and artificial means of vegetative propagations. Though both methods involve the development of a plant from parts of a single mature parent plant or part, the way that each is carried out looks very different. Let's talk about the artificial vegetative propagation. It is actually a type of plant reproduction that involves by human intervention. The most common types of artificial vegetative reproductive techniques include cuttings, layering, grafting, sucking, and tissue culture. These methods are employed by many farmers and horticulturists to produce healthier crops with more desirable qualities. Natural vegetative propagation is actually the method in which plants grow and develop in natural environment, in natural ecosystem, naturally, without any human propagation or involvement. So the vegetative propagation, which is in which plants propagate or multiply without any human involvement, is natural vegetative propagation. But with human involvement, it is the artificial vegetative propagation. So difference between natural and artificial vegetative propagation is this. So natural vegetative propagation is actually the very important ability of plants, where natural where that is the key to enabling natural vegetative propagation in plants is the ability to develop by adventitious roots. Adventitious roots means the roots which is developed by any parts. Though the formation of adventitious roots, new plants may sprout from stem, roots or leaves of a parent plant. Modified stems are also one of the source of vegetative plant propagation, like vegetative plant structure that arises from plant stem include number one, rhizome. Example is ginger. Runners, you can see here, runner, one part of running stem can be cut to grow naturally in new plants. Bulb of onion bulbs can be grown as new individuals. Tubers in potato, tubers form many new plants from one potato. Corms, these are the examples of natural vegetative propagation or vegetative propagation. If this method or this part can be developed by the activity of human involvement, it is artificial vegetative. But it is occur naturally without any human intervention, it is natural propagation. So tubers can also start from roots, and plantlets emerges from plants leaves naturally. Some vegetative parts by which plant propagation can be possible, we will discuss at first. So let's start. You should know some vegetative parts of the plants, like rhizome, which can be easily used for many species of multiplication by vegetatively. Rhizome is actually vegetative propagation, may occur naturally through the development of rhizome. Rhizomes are actually modified stems that typically grow horizontally along the surface or of beneath the ground. So it is a horizontal modified stems that remains in the soil. So rhizomes are storage site of growth substances such as proteins and starches. As rhizome extends, roots and shoots may arise from segments of the rhizomes and develops into new plants. So certain grasses like lilies, iris, orchids propagates in this manner. Edible plant rhizomes include like ginger and garlic. So in this picture you can observe a horizontal structure, horizontal underground rhizome, underground stem, that is called rhizome. In above picture you can see the small sucker is developed. Small sucker is developed, and this sucker can then develop into new baby plant, and root is developed from the lower part of the rhizome. Bulbs. Bulbs are actually rounded swollen parts of a stem. These are the modified underground stem, typically found in underground soil. Within these organs of vegetative propagation lies the central shoot of a new plant. Bulbs consist of a bud that is surrounded by layers of fleshy scale-like leaves, like an onion. These leaves are a source of food storage, and provides nourishment to the new plant. Example of plants that develop from bulb include onions, garlic, shallots, hyacinths, daffodils, lilies, and tulips. Corms are underground storage organs that are enlarged bulb-like underground stems. This vegetative structure store nutrients in fleshy, solid stem tissues and are typically externally surrounded by papery leaves. Due to their physical appearance, corm and commonly confused with bulbs. Bulbs present in onion is more like corm. The major difference between corms and bulb is the corm contains solid tissues internally, and bulb have only layers of leaves. Corm produces adventitious roots and processes buds that develops into new plant shoots. Plants that develops from corm includes crocus, gladiolus, and taro. These are the corm and remember onion. You can realize in onion bulb several layers of leaves are present, but in corm internal tissues are present. So this is the difference between corm and bulb. Runners. Runners are called stolons. Are similar to rhizome in that they exhibit horizontal growth at or just below the soil surface. Unlike rhizome, they originate from existing stems. As runners grow and they develop roots from buds located at nodes or their tips. Intervals between internodes are more widely spaced in runners than in rhizome. New plants arises at nodes where shoots develop, and this type of propagation is seen in strawberry plants and cruets. Let's come to discuss methods of vegetative propagation in detail. Layering is one of the method of or the technique of vegetative propagation, by which multiplication of plant easily propagate as new plants or multiplied as new plants. So stem is still attached to their parent plant and made from root where they touch a rooting medium. So it is the method of propagation a plant in which a shoot is fastened down to form root valve, and it involves bending plant branches or stem so that they touch the ground, or one part of the stem or branch go down into the soil, and where they are grow and as a new plant. So the portion of branch of branches or stems contact with the ground and then covered with soil, they adventitious roots or the roots that extend from structure other than plant roots develops in the plant covered by soils, and the attached shoot, branch or stem with new roots is known as the layer. So the type of layering, there are several types of layering is mentioned in this picture, like tip layering. The common example of tip layering is bougainvillea. Simple layering, you can see here a branch is go down into the soil where they form a root, and this is the simple layering. Then you can see here compound layering. Compound layering, for example, sometimes occurred in money plant. Several adventitious roots develops into new plants. If we rotate the adventitious roots into the soil, every new adventitious root develops into new roots as new plants. Mound layering, air layering, you can see here, stolen or runners. These all are the examples of layering in which the shoot or branches develops into new plant. One of the another method of vegetative propagation is suckering. Suckering by suckers. What is meant by suckers? It is very important to know what is sucker, and then you can understand the process of suckering. Suckers are actually vigorous vertical growth coming from the root or lower main stem of a plant. You can see here the vertical growth coming from the lower main stem of a plant. So suckers are usually considered undesirable plant branches, and because they sap the plant's energy. So suckers attach to a parent plant and form a dense compact mat-like structure, and since too many suckers can lead to smaller crop size, excess number are pruned, and mature suckers are cut away from a parent plant and transplanted into a new area where they sprout new plants. Suckering has the dual purpose of growing new shoots and removing nutrient sucking buds that prohibits the main plant from growing. So this is one of the important method of plant propagation. Some of the example of suckers are apple, elm and banana trees. The another very advanced and well-known technique of plant propagation is tissue culture. Actually, the tissue culture is a method of biological research in which fragments of tissues from an animal or plant are transferred to an artificial medium in which they can continue to survive and function. The cultured tissues may consist of a single cell, a population of cells, or a whole part of an organ. So this technique involves the culturing of plant cells that may be taken from different parts of a parent plant, and the tissue is placed in a sterilized container and nurtured in a special medium until a mass of cells known as the callus is formed. Callus is actually the part of tissue from where new part organ can be developed. So the callus is then cultured in a hormone-laden medium, artificial environment, and eventually develops into plantlets. When planted, this mature into fully grown plants. This is today considered a very common and popular technique of vegetative propagation. Plantlets are vegetative structure that develops on some plant leaves. This miniature young plants arises from meristem tissues located along leaf margins. Upon maturity, plantlets develops into roots and drop from leaves. They then take root in the soil to form new plants. An example of a plant that propagates in this manner is clanchu, or mothers of thousands. Plantlets may also develop from the runners of certain plants such as spider plants. You can see here on the edges of leaves there are many new plants grow with roots. One of the plants is detached from these leaves and can grow as new individual. So this type of plants are also called mother of thousands. Grafting. It is considered an old method which is applied in horticulture and gardening. This method is a process of joining two plants together, upper portion and lower portion, to grow as one single plant. So joining two parts from two or more plants so that they appear to grow as a single plant is the process of grafting. In grafting, a desired cutting is called scion. Scion is attached to the stem of another plant. The part of the stem of another plant is called rootstock that remains rooted in the ground. The tissue system of the cutting becomes grafted into or integrated with the tissue system of the base plant over time. So the best example of grafting is rose. What are the advantages of grafting? It is important to know what is the advantages. Actually, there are many types of grafting depending on size and position of scion stem, and the type of cut is made on stalk and scion. There are different types of grazing wise: approach grafting, side grafting, splice grafting, saddle grafting, flat grafting, and cleft grafting. So grafting is actually the fastest way of growing popular desirable varieties of fruiting trees and flowering shrubs on a large scale. Many commercially valuable plants are difficult to grow by other propagation methods like cuttings and layerings, but they respond well to grafting. Scion are highly precious varieties, but they usually have poor rooting system, less vigor, and weak defense against pathogen. All these flaws can be overcome by the process known as grafting them on the stalk, which is usually a hardy wild relative of a scion. Grafting is a way of cloning a scion variety to maintain the consistency of characters such as size, color, flavors, etc. With gardeners' curiosity, multiply varieties of different scions can be grafted on single stalk plants. Trees can be given ornamental shapes, etc. So different fruiting trees like mango, guava, chiku varieties can be grafted easily, and many flowering shrubs like roses, hibiscus, bougainvillea varieties can be grafted easily. You should know some vegetative parts of the plants like rhizome, which can be easily used for many species of multiplication by vegetatively. Rhizome is actually vegetative propagation may occur naturally through the development of rhizome. Rhizomes are actually modified stems. that typically grow horizontally along the surface or off beneath the ground. So it is a horizontal modified stems that remains in the soil. So rhizomes are storage site of growth substances such as proteins and starches. As rhizome extends, roots and shoots may arise from segments of the rhizomes and develops into new plants. So certain grasses like lilies, iris, orchids propagates in this manner. Edible plant rhizomes include like ginger and garlic. So in this picture you can observe a horizontal structure, horizontal underground rhizome, underground stem that is called rhizome. In above picture you can see the small sucker is developed. A small sucker is developed, and this sucker can then develop into new baby plant, and root is developed from the lower part of the rhizome. So students, thank you so much for being with me in this lecture. In next lecture we will meet inshallah. Allah Hafiz.